in your heads, and in your hearts. You have the power to try to become engaged in your own impacts on the planet. My name is Kat, and this is Sarah and Sophie, and today we'll be talking about the iTESLA-SoundBio 2018 iGEM project, Document the Difference. In today's presentation, we'll begin by going over the background and purpose of our project. Next, we'll go over our design and goal and our results. And finally, we will end with our future and the impact we hope to make.
So project background and purpose. Our project deals with detection of endotoxins. Firstly, let's talk about why endotoxins are bad. So when endotoxins are injected into humans through a drug, for example, they will induce a large immune response. This may result in fever, inflammatory response, shock, organ failure, and even death. So how can we detect this? Well, horseshoe crabs have a unique ability to detect endotoxin through their blood. This is extremely valuable to the biomedical industry because they're able to use this quality of horseshoe crab blood in order to make the LAL test. The LAL test can detect endotoxin in drugs and hospital equipment. However, horseshoe crab blood currently goes for around $15,000 per quart, and it poses ecological consequences as well. The harvest of horseshoe crabs for their blood is extremely detrimental to horseshoe crab populations, and in turn affects marine ecosystems as a whole, because animals such as turtles and birds such as red mouse rely off of these animals for sustenance. So, some facts about why this population decline is happening. It takes around 250,000 horseshoe crabs to make 1 vial of LAL, and during the harvest process, more than 30% of horseshoe crab blood is drained. This causes around a 20 to 30% mortality rate among horseshoe crab populations, and in the long term, it can take up to 10 years for horseshoe crabs to recover back to their original state. So a little bit about the harvest process. The horseshoe crabs are collected. They're stabbed in the tissue around their heart to extract blood. Then amoebocytes are extracted from their blood. These amoebocytes hold clotting factors that can trap contaminants in a gel-like substance. After the amoebocytes are extracted, the horseshoe crabs are then returned back to the wild. And though biomedical experts like to believe that these horseshoe crabs are able to successfully integrate back into the wild, this often isn't the case.
To get an inside look on horseshoe crab harvesting, we decided to reach out to experts. One of the first experts we contacted was Dr. Thomas Nowitzki, the retired president and CEO of Associates of Cape Cod, one of the very first companies to ever work with LAL. From Dr. Nowitzki, we learned that in recent years, regulations have been passed in the U.S. to curb horseshoe crab harvesting, and therefore it is currently deemed as sustainable. However, from Dr. Nowitzki, we also learned that this is not the case in international countries. For context, the 4 subspecies of horseshoe crabs, as listed on the slide, are found in 2 main regions, the Atlantic coast as well as in Asia. And to learn more about how international regulations are impacting horseshoe crab harvesting, we decided to reach out to Dr. Ng Wai Chen of Hong Kong's Agriculture, Fisheries, and Conservation Department. From Dr. Ng Wai Chen, we learned how horseshoe crab harvesting has directly impacted the ecological diversity of Hong Kong's marine ecosystems. Not less than 10 years ago, the Hong Kong shores have all 3 species of Asian horseshoe crab. However, due to harvesting, now they only see 2 of the subspecies, and this number is currently and fastly declining. Additionally, from Dr. Ng Wai Chen, we learned how countries like Hong Kong are currently taking steps to curb horseshoe harvesting. However, this is not the case in other Asian species.
for that organism. And after we did that, we realized that this gene sequence was over the limit that IDT had for a gene block. So we then had to split our into 2 fragments. And once we received our 2 fragments, we then individually digested and ligated each one into their own TSD1C3 backbone, then transformed it into E. coli for further cloning, thereby increasing our yield. And once we have cloned fragments, we then digested both with BIGL2 and TSD1. And thereby, we would be isolating fragment 2 from its plasmid and creating the necessary CDN after fragment 1. So in the end, once you ligate the 2 together, you then get the whole entire factor C sequence inside the TSD1C3 backbone ready to be shipped as a biobrick. Moreover, we want to do further experimentation with this protein, so we also integrated into the PAXA1 bacillus integration vector as well.
So our design of phase 1 is greatly influenced by the work and the information that we received from experts that we contacted. One of the first people we reached out to was Dr. Daniel Ziegler, the director of the Bacillus Genetics Stock Center and professor at Ohio State University. From Dr. Ziegler, we learned that Bacillus subtilis would be a better organism than B. cereus to host our factor C. Additionally, he suggested that we use a specific strain of 168. And it was from Dr. Ziegler that we learned that PAXA1 would be a suitable integration vector for our project. The other expert we contacted was Dr. Karen McClymont. We first learned about Dr. McClymont from her PhD thesis on LAL assay, and so we reached out to her to learn more about the troubles and obstacles she had when she was doing her work. One of the main things that she talked to us about was how factor C has high sensitivity to LPS, or lipopolysaccharide, a molecule found in the membrane of gram-negative bacteria. And in addition, Karen McClymont also talked to us about how LPS contact leads to protein clotting.
To clarify, factor C only cleaves in the presence of endotoxin. It itself does not indicate the presence. It only cleaves once it's exposed to it. So our project sought to directly detect cleaved factor C. And in order to do so, we considered 4 main options. The first option would be to synthetically produce all natural proteins that are in the naturally occurring LAL cascade. This should work as it's supported by previous works in industry and academia, but it would be challenging for a high school team like us in terms of money and time needed and money spent on reagents. The second option we considered would be to identify a new zymogen, an inactive enzyme precursor to react with cleaved factor C. This is currently used in practice by biomedical industries by using a fluorophore peptide, similar to factor B that reacts with cleaved factor C. While this does make the LAL cascade a lot more simple and time-effective, critics have stated that by cutting out the intermediate steps of the LAL cascade, recombinant factor C makes it lose its specificity. The third option we considered was using disopropyl fluorophosphate, DFP, as a substrate to react with cleaved factor C. This should work because in literature we read that cleaved factor C has a site that's sensitive to DFP. And to get some more tips and advice on how we could carry out this option, we contacted Dr. Thomas Nowitzki, who warned us in his response that DFP is actually extremely toxic to human beings as it can cause intense eye twitching, ciliary body spasms, and headaches as well. So with that option tossed aside, then that brings me to our final and most promising option, using a Western blot to compare uncleaved factor C with sample exposed factor C. This should work in theory because uncleaved factor C is 123 kilodaltons, whereas cleaved factor C has 2 parts. One is 80 kilodaltons and the other is 43. So if a sample were to have endotoxin and it were to be compared on the Western blot, the sample exposed factor C should be cleaved and thereby appear shorter than uncleaved factor C. We also designed 2 biobricks that we're planning on submitting for this year's season. The first one we planned on submitting would be of the factor C sequence that's codon optimized for Bacillus subtilis. The second biobrick we had in plan was of the PAXA1 integration vector along with the xylose cassette, which contains a xylose-inducible promoter. With these plans and designs set in place, we spent the rest of our iGEM season putting these plans into action. The first part of our project addressed synthetically producing factor C inside Bacillus subtilis. While we weren't able to do so because we had issues and difficulties in cloning and PCRing, we were able to fully codon optimize the factor C gene sequence was to create a BioBrick compatible Paxa1 integration vector. Also had similar issues with PCRing. However, we did realize that the whole sequence of Paxa1 and Xylus cassette were both together and ready to submit. However, there were some illegal restriction sites. So we turned to site-directed mutagenesis to either insert or delete nitrogenous bases without altering the function of the sequence. And lastly, we had to experiment with Bacillus subtilis as well, as this was our first year working with this type of chassis. So we experimented with 5 different transformation protocols, one of which gave us results, but low efficiency transformations. And to make sure that our plasmid not only transformed, but also integrated into the Bacillus genome, we designed 6 sets of primers to make sure that the whole thing integrated into the genome. While we again had issues with PCRing and colony PCR, that led us to believe that perhaps the cells we were working with were not Bacillus subtilis, but rather contamination that found its way onto the agar plate. So instead of focusing our primers on colony PCRing, we decided to focus on the primers to see whether we had the right genome of Bacillus subtilis. And after further blast sequencing, we can confirm that the cells we were using were the right strain.
Now will we go over the project future and the impact.
So our project holds 2 main impacts, environmental and in industry. So ecologically, by synthetically producing factor C, we can help protect horseshoe crab populations from harvest and promote global ecosystem health. Additionally, through future applications, we can help to create a more accessible endotoxin detection test by lowering the cost of our final product, as well as shed light on the possibility of ecologically conscious biomedical advancements.
So to elaborate on our future plans, we would follow a 3-step process as outlined on the screen. Firstly, we hope to perform rigorous testing on our endotoxin test to ensure that it does not create a pyrogenic response and does not give us any false positives or false negatives. The second thing we would focus on would be gaining FDA approval, which typically takes 10 to 12 years. And finally, we would work on implementing our drug test internationally. And hopefully with the international implementation of a synthetic LAL assay, we will be able to alleviate the detriments of current horseshoe crab harvesting. To elaborate further on FDA approval, we contacted a few specialists. From one of the specialists, Dr. David Husong, who works at ValSource and wrote an extensive article on the LAL assay, we learned about what happens to synthetic biology alternatives that try to seek a place in the market. Dr. Husong detailed that any synthetic alternative to a drug or a drug test needs to be just as precise and just as effective, or more effective, than the current standard. Additionally, Dr. Husong taught us that if we wanted to implement our drug test internationally, it would have to go through international testing by the 3 major pharmacopoeias, the US, Japan, and Europe. The second specialist we talked to was Ms. Brenda Rose from the Drug Division of Information at the FDA. Ms. Rose informed us that our test would have to go through what's called pyrogen and endotoxin testing, which ensures that our test does not let a pyrogenic response occur, and this would happen at the Center for Drug Evaluation and Research. Another significant aspect of the iTESLA-SoundBio project this year was the educational impact that we tried to make on our community. One of the first things that we did was hold an outreach event at Gasworks Park. At Gasworks, we work with young individuals in our community to teach them about DNA, as well as the impact of synthetic biology in our community. Another event we held was at the Seattle Mini Maker Fair, where we worked with our community lab, SoundBio, to set up a booth for the citizens of Seattle. I had the pleasure to talk to individuals about how LAL has played a role directly in our community, namely in Seattle-based companies such as Rim Bio, as well as the Seattle Children's Hospital. And as I mentioned, the iTESLA-SoundBio team is unique, as it functions out of a community lab. And that is why the Baltimore Bio Crew, a high school iGEM team that also functions out of a community lab, reached out to us to create a 6-section guide for community labs all over the world who are also interested in participating in high school iGEM. This 6-section guide covered topics from fundraising to recruiting and was made possible by the responses from 11 community labs from around the world. This 30-page comprehensive guide can be found on our wiki to download and share. And finally, the iTESLA-SoundBio team sought to learn more about how individual action plays a role in creating ecological change. One of the first organizations we contacted was the IUCN Horseshoe Crab Specialist Group, spearheaded by Mark Botton and Paul Shin. The IUCN Horseshoe Crab Group has been extremely influential in creating new regulations in the US for decreasing the import of Asian horseshoe crabs. And in addition, the IUCN Horseshoe Crab Group was able to add the 3 Asian horseshoe crab species to the IUCN Threatened Species Red List. Another individual we contacted was Tiffany Black from the Florida Fish and Wildlife Conservation Commission. From Ms. Black, we learned about the importance of contacting legislators and other organizations to raise awareness about ecological issues such as horseshoe crab harvesting. And additionally, from Tiffany Black, we learned about the importance and significance of individual action in our community. To end, while we did talk about how the iTESLA-SoundBio team tried to synthetically produce Factor C, we hope to leave you with this. For any ecological change to occur in our world, it needs to stem from the cooperation of individuals and the dedication of our peers. And that is why, as Tiffany Black said to us, education helps people become more involved in protecting wildlife. Because with education comes appreciation. And with appreciation comes protection. And this is something that we in agencies strive to do every day.
Our project would not have been possible without the contributions of our PIs and team advisors, as well as many others that helped us with our project. Thank you, and do you have any questions?
Thank you guys for a great presentation.
Sorry, can you go back slides to the part you mentioned the Western Blot? Yeah. Okay, in that part you mentioned that the difference was the cleavage, right? Was the size. Why did you decide to make the Western Blot instead of just a gel?
So, to our understanding, the Western blot is a type of gel that's specifically designed for proteins. For further elaboration to make our idea more clear, this would work by adding a his tag to the factor C protein at only one side. So then, depending on where the his tag is located, then we can determine where the cleaved factor C would appear on the gel.
I was very impressed with the thoroughness of your background research. It's a really good job on that. So if you have the chance to come back and continue on this project next year, what are your next steps specifically?
So, one issue we had this year was the cloning process itself, and we actually, late into the iGEM season, we found other options, such as we wanted to experiment more with fast cloning and perhaps Gibson assembly, as we see that that's probably the most effective option that other iGEM teams use.
Additionally, we also wanted to work on, as we mentioned earlier, a cheaper and effective test. The current LAL assay, as Sarah mentioned, goes for $15K a quart, and we hope that we will be able to create a more portable and effective means for testing the sterility. Because in third world and developing countries, SSRIs, or surgical site infections, are extremely prevalent, and we hope that the LAL assay test or an endotoxin test is useful.
Really quickly, we would also run some cleanups and just diagnose what's going on in our regular protocols, such as debugging our PCR protocol.
Can you guys talk about the motivation behind the PAXA1 integration?
So we got the
idea for the PAXA1 vector from contacting professors in our research who suggested to use PAXA1 with Bacillus subtilis, Dr. Zieler specifically. We also wanted to use an integration vector with Bacillus because Bacillus subtilis 168, the strain we were working with, is known to frequently pop out plasmids, so we wanted to make sure that the gene is integrated into his genome.
Thank you again. Great job on your presentation. Everyone, a round of applause.
All right, that takes us to the end of this session. So if we can everyone clear out the room, the judges will need to meet and confer. Thank you.
High five. High five. High five.